Hi there. I'd like to take a few minutes and show you the characteristics of some bacteria species grown on trypticase soy agar plates by students here in the lab and see how those bacteria are behaving on trip soy agar. So let's take a few minutes and take a look at a few bacteria. What I have in front of you here is Serratia Marcescens. The interesting thing about this colony or this plate of Serratia is it has bright red colonies. This one was grown at room temperature. You can notice the RT stamp on there for its entire life. And that room temperature growth, especially the start of growth, tends to give Serratia red colonies. If you'll notice this plate, this is also Serratia, but this one was started in the incubator and has then since spent quite a few days, this one about 5 days, at room temperature. So even though it's been at room temperature for a long period of time and it came from exactly the same starting broth as this one, it's still not making red colonies. So it's worth being aware that for Serratia to produce red colonies, it's better to actually start it at room temperature, not start in the incubator and then move to room temperature. Another colorful colony we can take a look at is this one. This is Micrococcus Luteus. Luteus makes these bright lemon yellow colonies that tend to be round, a little smooth. This one's a little bit old and they're starting to flatten out in the middle, so we've got some with raised edges on a slightly older colony. This happens to be Pseudomonas aeruginosa, and I don't know if you can tell it or not here, but Pseudomonas tends to give kind of a green tint to its media. It's not always very apparent, and it's not strikingly apparent in this case, but it does. This tends to have a colony that has spreading edges. I don't know if you can see the detail on the colony right here, but it's denser in the middle and spreading out on the edge. In some cases a little bit umbonate, meaning it's got kind of a tall center, almost like a fried egg out on the plate. Next, we're going to take a look at Ecoli. This looks like many bacteria. It's got sort of whitish, creamish looking colonies that are more or less round, maybe with slightly irregular edges. Nothing particularly striking, although it does have a distinctive funky odor when you open the plate up. Here we have Bacillus cereus, the infamous white, fuzzy, kind of spreading colony that tends to overrun other things on a plate. That would be Bacillus cereus. Next we have two together, two that are related. These are both Pseudomonas. They're Pseudomonas mirabilis. I'm sorry, I misspoke. It's not Pseudomonas, it's Proteus. Proteus vulgaris and Proteus mirabilis. And the interesting thing about Proteus mirabilis is it spreads. There are bacteria all over this plate, and I don't know if you can see the source, but one single touch right in the center is all that was done to this plate, and it spread out and covered the entire plate. This one swarms like crazy. But probably more important on both these Proteus plates, if you notice the agar has got kind of an amber orange tint to it. That did not start out that way. This was like any other trypticase soy agar. It started out clear, and Proteus tends to make its agar this amber brown color. A great tip that you might have Proteus. This one happens to be Lactococcus, and it doesn't look like there's much going on the plate. In fact, this colony right here at my fingertip is actually a contamination that is not Lactococcus. But if you look closer, right in the area of my thumb, there are some small, round, white colonies that are Lactococcus. Let me see if I can focus on that spot. And that would be Lactococcus. Next we have Citrobacter frundi. Citrobacter, some obvious white colonies that are slightly irregular, a little bit umbonate with a tall center. Citrobacter. Here we have Alcaligenes faecalis. We've got a pretty aggressive grower here. It has irregular colonies. A little bit of coloration maybe to the media, but we have kind of a mostly white to cream color irregular colony for ecologenes. Here we have Staph Aureus. The word aureus refers to the chemical symbol for gold, which is AU. This is sometimes called golden staph, and while it's not as bright lemon yellow as Micrococcus luteus, it does have kind of a yellowish tint to it. These are round, smooth colonies. This plate is about a week old, and you can see from my fingertip how big these colonies are. That would be Staph Aureus. This looks like Staphylococcus Epidermidis, and it is. And the thing to notice about Staph Epidermidis is we have, again, small white round colonies that are smaller than the other staph that I just showed you. Here is Staph Aureus. Here's Staph Epidermidis. Nice, tight, small white colonies that are round and smooth. Convex, by the way, as well. Shigella flexneri. Similar to what we just saw with Staph Epidermidis, we have small, round colonies. They're a little bit more translucent, kind of a creamy, not quite clear color than the Staph Epi we just saw. And as I tilt the plate there, you can get a little bit better look at the overall shape of those colonies. And the last one I'm going to show you today would be Kludgy Ella. This happens to be Kludgy Ella pneumoniae. We have a more cream-colored, again, round, smooth, and convex shape. These that are about a week old tend to be a little bit tall in the middle, so ever so slightly ambenate. Hopefully that'll help you in identifying some of your microbes. Thanks for watching.